Welcome back to Chapter 7, Carbohydrates and Glycobiology. In this lecture, we are going to look at Part 2 of this chapter. Here, we will learn about disaccharides, oligosaccharides, and polysaccharides. What are disaccharides? Disaccharides are two monosaccharides joined by a covalent linkage known as O-glycosidic bond. Some of the examples are sucrose, lactose, and maltose. Sucrose is a combination of glucose and fructose joined together by a glycosidic linkage. Lactose is between galactose and glucose by a glycosidic linkage. Maltose is a dimer of two glucose molecules, again formed by a glycosidic linkage. Two sugar molecules are joined between an anomeric carbon and a hydroxyl carbon. As you can see, the anomeric carbon of the left sugar, and this is the anomeric carbon, is joined with another carbon that can be an anomeric carbon or a different carbon with a hydroxy group. A disaccharide that is shown here, like maltose, is formed from two monosaccharides, alpha D-glucose and beta D-glucose, when a hydroxy group of one of the monosaccharide condenses with the intramolecular hemiacetal of the other, that is shown here. This results in the elimination of water and formation of a glycosidic bond. This is called as a glycosidic bond. And this is not a hemiacetal, but an acetal. One of the major difference between a hemiacetal and an acetal is that hemiacetal is a reducing end and acetal is a non-reducing end. Now you have to remember that in maltose, there is one hemiacetal and one acetal group. Let us see as to how we can name disaccharides. First, name the configuration, alpha or beta, of the anomeric carbon of the first sugar. Let us consider an example. Lactose. In lactose, which is made up of beta D galactopyranose and beta D glucopyranose, the first sugar is beta D galactopyranose. And as I said the name, beta is the configuration of the anomeric carbon. Now, since this is a glycosidic bond, a beta configuration is given because the hydroxy group is above, and that is what is forming a glycosidic bond with the 4 carbon of beta D glucopyranose. Next, add pyranosyl or furanosyl to distinguish 6 and 5 membered rings. In case of lactose, it was 2 6-membered rings, and hence pyranose is used. In case of sucrose, which is a combination of glucose and fructose, one uses fructofuranosyl and alpha D glucopyranosyl. Indicate the 2 carbon atoms joined via the glycosidic bond in parentheses. In case of lactose, the 2 groups that are joined, the glycosidic bond that is joined happens between beta, that is the anomeric carbon, and hence beta 1 to 4, and in this case is the 4, and hence in parentheses you show beta 1 4. In case of sucrose, it's either the glucose alpha 1 to 2 beta to fructose. In case of glucose, it's the first carbon, and in case of fructose, it's the second carbon. But remember The second carbon is the anomeric carbon for fructose, and the first carbon is the anomeric carbon for glucose. And hence in sucrose, which is a non-reducing sugar, there is no hemiacetal group left, whereas there is a hemiacetal group in lactose. Name the second residue using the same pyranose or furanose form. In case of trihalose, you first name the first sugar and then the second sugar. It could be either pyranosyl or pyranoside, depending on whether it's a 5-membered or a 6-membered. And pyranoside, because it's the glycosidic bond is attached to the anomeric carbon. Repeat the process if there's a third sugar. So these are the 3 examples that I've used in naming these disaccharides. And all these 3 are disaccharides. Remember, if you have a 1-1 linkage as shown here, gluc alpha 1 to 1 alpha gluc, this means that this only has an acetal group. There's no hemiacetal group because there's no other anomeric carbon, as unlike lactose. Lactose has 1 anomeric carbon and hence it is a reducing sugar. Sucrose is a non-reducing sugar, unlike lactose, because no anomeric carbons. Again, reiterating my point, the anomeric carbon in the first saccharide is an acetal or a ketal, thus non-reducing. This is an acetal. Because this is an acetal, this is non-reducing. If the second anomeric carbon is free, it can still linearize into an aldehyde or a ketone, and hence this hemiacetal is reducing. Polysaccharides. Polysaccharides are long sugar polymers. Homopolysaccharides are sugar polymers that have one type of monosaccharides. Heteropolysaccharides have different polysaccharides. Polysaccharides can be linear or branched. They are typically used in structural or storage roles. Here is a slide depicting the differences between homopolysaccharides and heteropolysaccharides. Now, homopolysaccharides, as I said, can be unbranched or linear or branched. And as shown by the color of these cyclohexagon groups, it says that all these monosaccharides are the same. It could be arranged in a linear fashion or in a branched fashion. The same way, heteropolysaccharides could be made up of 2 monomer types in this specific case, or more. And in case of a 2 monomer types, unbranched, it's shown like this. Or it could be multiple monomer chains which are branched. Glycogen is an example of a polysaccharide. Glycogen is a branched homopolysaccharide of glucose. Remember that, a homopolysaccharide of glucose. Glucose monomers form 1,4 linkages, as shown here. This is 1, and in this case, this is 4. So 1,4 linkages, that's a glycosidic bond. Branched points with alpha 1,6 linkages. So this is alpha 1,4 linkage because this hydroxy is pointing down, as shown here. And alpha 1,6 linkage is formed because this is 1 and this is 6. And alpha 1,6 linkage is the branched, that is every 8 to 12 residues in case of glycogen. Molecular weight of glycogen reaches several millions. They function as the main storage polysaccharide in animals. Starch is another example of a polysaccharide. It is different from glycogen in the sense that starch is a mixture of two homopolysaccharides of glucose. What are those? Amylose. Amylose is an unbranched polymer of glucose. It has alpha 1,4 linked residues. Amylopectin is a branched polymer like glycogen, but the branch points with alpha 1,6 linkers occur every 24 to 30 residues, unlike glycogen. So starch is a mixture of both amylose and amylopectin. Molecular weight of amylopectin is up to 200 million. Starch is the main storage polysaccharide in plants. This is how starch looks like. Amylose, as I said, is a linear polymer of glucose. It's alpha 1,4 linkage as shown here, 1,4. Amylopectin has branches, alpha 1,6 branches every 24 to 30 residues. Unlike glycogen, which has more branches every 8 to 12 residues. Now, starch granules has a combination of amylose and amylopectin. And as you can see, these branches in amylopectin are shown here. This 1,6 branch is shown here as this red node. And additionally, there are reducing ends as well in starch where there is amylose or amylopectin. Remember, reducing end exists because there is a free anomeric carbon with a hydroxy. As I said before, glycogen is a polymer of glucose, alpha 1,4 glycosidic linkages. And every 8 to 12 residues, there is a branch point in glycogen. And in addition, it also has one reducing end and many non-reducing ends. Now, did you know that glucose in our body is stored as glycogen? Why can't glucose stored in monomeric form? It is believed, and there is scientific evidence stating that about 0.01 micromolar or 10 nanomolar of glycogen can break up into 400 millimolar glucose. That's a lot of glucose. Now, why would you want to store glucose in the form of glycogen? Is because of the consequences that arise due to a very high concentration of glucose inside a cell. 400 millimolar glucose is a very high concentration inside the cell. And what are the consequences? Elevated level of glucose can result in high osmolarity. Now remember, an increased osmolarity would be threateningly elevated, and that can lead to entry of water, leading to the rupture of the cell. Now it can also lead to transport of glucose, and glucose transport is essential, and we do not want glucose to get out of the cell and enter other cells, and hence glycogen is stored as a polymer of glucose in liver, and only when there is a requirement of glucose will the glycogen molecule get broken down by enzymes to individual glucose molecules. So this is a classic example as to how polysaccharides play a role in our body. So what is the metabolism of glycogen and starch? Glycogen and starch often form granules in cells. Granules contain enzymes that synthesize and degrade these polymers. Glycogen and amylopectin, both branched chain polymers of glucose have one reducing end, but many non-reducing ends. Enzymatic processing occurs simultaneously at many non-reducing ends to release glucose. Now let's look at some structural polysaccharides. Cellulose is a structural component of plants. They are also composed of amylose and amylopectin, but the linkages are beta, not alpha. Please remember the difference. Chitin is a homopolymer of N-acetylglucosamine, or GlycNAc in short, in beta linkages. And chitin is found in the exoskeleton of insects. Cellulose is another example of a branched homopolysaccharide of glucose. Cellulose is composed of glucose monomers that form beta-1,4 linked chains, as shown here, beta-1,4. So this is beta-1,4 linked D-glucose units. The difference between cellulose and amylose is that amylose is alpha-1,4 linked. Hydrogen bonds form between adjacent monomers, as you can see. Additional hydrogen bonds between chains. Structure is now tough and water insoluble because of these hydrogen bonds. And cellulose is the most abundant polysaccharide in nature. Cotton is nearly pure fibrous cellulose. Let us look at cellulose metabolism. The fibrous structure and water insolubility makes cellulose a difficult substrate on which to act. Fungi, bacteria, and protozoa secrete cellulase, which is an enzyme. This allows them to use wood as a source of glucose, because cellulase can convert the cellulose in wood into glucose. Most animals use cellulose as fuel source because they lack enzymes to hydrolyze beta-1,4 linkages. Example is us. We do not have cellulase in us, so we cannot eat wood. Ruminants and termites live symbiotically with microorganisms that produce cellulase. Cellulases hold promise in fermentation of biomass into biofuels. Chitin is a linear homopolysaccharide of N-acetylglucosamine. As I said before, chitin forms the protective covering or the exoskeleton of insects. N-acetylglucosamine monomers form beta-1,4 linked chains, and they form extended fibers that are similar to those of cellulose. The difference is that chitin is hard, insoluble, and cannot be digested by vertebrates. The structure is tough but flexible and water insoluble. It's found in cell walls in mushrooms and exoskeletons of insects, spiders, crabs, and other arthropods. Polysaccharides have higher order structures. For example, disaccharides can rotate around the glycosidic bonds. The monosaccharide ring acts as planes that rotate around the point of the glycosidic oxygen. Therefore, they have phi and psi angles. How surprising. We are revisiting the Ramachandran plot here because monosaccharides and disaccharides can rotate around the glycosidic bonds. Hydrogen bonding between residues is also important for the structural integrity of a polysaccharide, as we saw in case of cellulose. When do we see alpha versus beta linkages form? Structural polysaccharides often utilize beta linkages rather than alpha. Please remember this. In amylose, for example, with its 1,4-glucose linkages, the most stable configuration is a 60-degree bend between the planes, as shown here. This limits the hydrogen bonding availability between sugar residues and thus promote a loose alpha helical structure like this. So the bottom line is, like peptides, sugars also have secondary structures. And in case of amylose or polysaccharides with alpha linkages, they are bound to form loose alpha helical structure because they have limited hydrogen bonding availability between sugar residues. In cellulose, however, with its beta 1,4-glucose linkage, the most stable configuration is a 180-degree bend between the planes. This readily allows hydrogen bond to form between monomers and across strands, and as a result allows for this kind of a hydrogen bonding network. Shown in this figure is a Ramachandran plot for this disaccharide gal beta 1,3 gal. Now the plot shown here is a three-dimensional plot. As you can see, psi is plotted against phi, and the third dimension represents the relative energy. And as you can see, a peak means a high energy, and a plateau means a low energy. And what this plot tells you is that for a molecule like a disaccharide such as gal beta 1,3 gal, the most favored conformation is when phi and psi is about 30 degrees and minus 40 degrees, which looks like this. This involves the maximum hydrogen bonding ability. Whereas a phi and psi value of minus 170 and minus 170 is least favored for this molecule. Certain marine red algae, including some of the seaweeds, have cell walls that contain agar, which is a complex mixture of heteropolysaccharides containing modified galactose, as shown here. This modified galactose has sulfates, and they serve as the units of cell wall. Polysaccharides are also components of bacterial cell walls. The cell walls of bacteria are made up of cross-linked heteropolysaccharides. Peptidoglycan, as it is called, is made up of strands of alternating N-acetylglucosamine that makes glycosidic linkage beta 1,4 with N-acetylmuramic acid. Cross-linked to protein scaffold via murnac residues or muramic acid residues. And the result is that the cell, bacterial cell, is resistant to osmotic pressure and it gets a cell structure. Here is an alternating structure of glycnac and murnac, the two monosaccharides in the peptidoglycan layer. Now, as the name suggests, glycan chains are connected to each other via peptide bridges, and hence the name peptidoglycan.